Hi everyone, my name is Sasha,
And I'm Rachel.
and we're going to be talking a bit about the enzyme alkaline phosphatase.
So, what is alkaline phosphatase, also known as AP? It is a metalloenzyme, which means that it uses metal cations as cofactors, specifically in this case, zinc and magnesium. It is also dimeric, which means that it has two subunits and then they are connected by cysteine residues. And it is also part of the class of enzymes called hydrolases, which means that it catalyzes hydrolysis reactions, specifically in this case. It catalyzes the reaction of a phosphate monoester cleaved by water to make alcohol in a phosphate. And it is also a group of isoenzymes, meaning that it is present in various tissues and they all have different physiochemical properties, but they still facilitate similar hydrolysis reactions. So AP is commonly found in many organisms like yeast, E. coli, and mammals. In yeast, it is found in lysosomes similar to vacuoles. In E. coli, it is found in the periplasmic space, and in mammals, it is found as a glycoprotein on the cell membrane. So some primary isoenzymes specifically, there are four main types of mammalian isoenzymes. There is one in placental-like, which is like germline, intestinal, and tissue nonspecific, which used to be commonly known as like liver, kidney, and bone, just because it did so much. And after much research, they realized it encompassed a lot more. And this picture is a depiction of our protein that we made in Swiss model.
So following that brief introduction, here's an overview of the rest of our presentation. We're going to talk about alkaline phosphatase at the gene level, followed by the protein level. Then we're going to make some relations to existing research, followed by a mention of some practical applications that can be used in the lab.
So first, we're going to dive into AP at the gene level. So as we know, DNA is the template for all things in cells. So specifically, this is a picture of adult human intestinal alkaline phosphatase, and it is comprised of 1,587 nucleotides.
Multiple splice variants of this enzyme also exist. They're created through alternative splicing and the selective inclusion and exclusion of exons or coding regions of pre-mRNA. So this is a depiction of the multiple splice variants. As you can see, they have slightly different lengths depending on what exons are included. And then they have different isoform characteristics. Something we thought was interesting was that the isoforms that were labeled as pre-pro proteins are thought to be processed to generate what's known as the mature form of alkaline phosphatase.
So another tool we used is BLAST, which basically allows us to put the nucleotide sequence in and then compare it to the database's nucleotide sequence. So our sequence that we put in is also known as the query. And then the subject line is basically proteins that we are comparing it to in the database. So one of the ones that it is most similar to, but still has some differences, is the pan-peniscus. And it basically just shows us all the nucleotides and how they are similar, and then the gaps show us where they vary. So in our query, it is a guanine, and then in our subject, it is a thymine. And there are multiple gaps found within, and this is the continuation of the alignment sequence. And yeah. So now we're going to dive into AP at the protein level. So there are many types of common AP isoforms, which using BLAST again, we were able to see which proteins have the most nucleotides that are similar. And the first two are still found in humans. The third one is found in gorillas. And then the last three are found in different types of organisms that are very similar to monkeys. So BLAST also allows us to have a visual depiction of where the nucleotides are kind of different. So the red boxes kind of show us where the conserved domains are. And as you get further down the list, you can see how different amino acids that are formed from the nucleotide differences can lead to different folding patterns.
So switching gears a little bit, we're going to talk about some important subdomains or structures. First, we have the signal peptide of the enzyme. This is located in the N-terminus, and it guides the enzyme to the endoplasmic reticulum during the synthesis process. We also have the alkaline phosphatase domain, which is basically the location of the active site. We also have metal ion binding sites, which stabilize the enzyme and, of course, coordinate with the metal ions that are needed for catalysis. We also have glycosylation sites where the enzyme covalently attaches to carbohydrates. The locations of these vary by isoform. In some isoenzymes, we also have what's called a crown domain, which is a structural component of intestinal alkaline phosphatases that are sometimes found what could be known or referred to as the top of the enzyme, hence the name crown domain. Also in some isoenzymes are an N-terminal alpha helix, and as you would expect, this represents a helical structure at the N-terminus of the protein. We also predicted the secondary structure of the intestinal type AP using the platform listed here. So we entered the amino acid sequence for the enzyme, and we were shown where alpha helices in pink and then beta sheets in yellow are likely to form within the sequence. And then the Swiss model to the right gives a more comprehensive visual of that. We also simulated an NMR spectrum for the enzyme. This probably looks different from NMR spectra you've seen in previous classes. This is called a heteronuclear single quantum coherence spectroscopy, NMR, in which the shifts of the various amino acids within the protein are shown. It's similar to CNMR and HNMR, except that it's specifically used in protein studies, and this is for AP and E. coli specifically. These are also some protease cleavage sites that we found specifically for chymotrypsin and trypsin, and this is for the intestinal type AP precursor, and we found these using the XPASY peptide cutter. This is the same thing except for AP in E. coli. And then interestingly, we found that the predicted cleavage sites for AP and E. coli with trypsin were actually reported in the scientific study shown here. So looking at the sequence and then counting, we can see that the reported cleavage sites align with the predicted cleavage sites. It's important to also note that glycosylation is a really important post-translational modification that AP undergoes, as it's what gives rise to the various alkaline phosphatase isoforms that we discussed specifically. Both N-linked and O-linked glycosylation are at play here, and this is just a generic scheme of what N-linked glycosylation might look like at the cellular level. Moving on, now we can talk about the mechanism of the enzyme. So first we have the creation of the enzyme reactive oxygen phosphate. This involves the coordination of an ester oxygen to a zinc ion. Next, we have the formation of a covalent enzyme phosphate intermediate in which phosphate moves into the active site. Next, we have the formation of a modified enzyme phosphate intermediate that's formed when a nucleophilic attack occurs in an alkaline pH. Lastly, we have the dissociation of phosphate, as shown here. This is known as the rate determining step or the slow step of the reaction in alkaline conditions. We also predicted the molar extinction coefficient of our protein. This is the amount of light a substance can absorb at a specific wavelength. We estimated this using what's called the Gill and von Hippel method, which is briefly described here, and we ultimately calculated the extinction coefficient to be 56,620 inverse molarities times inverse centimeters at a wavelength of 280 nanometers specifically. We also predicted the PI, or isoelectric point, for the enzyme using the embossed PEPSTATS tool. For the intestinal type in humans, we found it to be approximately 5.7. And comparing these two enzymes, the lower PI actually makes sense given the more negative net charge, as you would need a lower pH to protonate maybe more of those negatively charged amino acids to result in that net zero charge, which is what the isoelectric point represents.
So now we're going to compare everything that we learned to existing research. So the article we read was Quinone Methide Signal Amplification Covalent Reporter Labeling of Cancer Epithelials Using Alkaline Phosphatase Substrates. So some background. Basically, this study was looking at different diagnostic tools that use immunohistochemistry assays to have diagnostic tools for cancer. And they basically are under this whole type of group where they used CARD, which is also known as catalyzed reporter deposition, and reporter meaning that it's just a molecule that is able to bind to tissue and be able to quantify and like stain what we're trying to look at. So the current tool that they use is HRP-based TSA, which is also known as horseradish-based tyramide signal amplification. And the way that this works is that tyramide in the presence of hydrogen peroxide becomes highly reactive with electron-rich amino acids and proteins, and then it is able to quantify where the binding of tissues are, cancer tissue. And then the only problem with this is that it's kind of expensive and uses a lot of chemicals because between each step, you have to inactivate the HRP or the horseradish peroxidase. So that's why this study kind of looks at another potential imaging tool known as AP-based QMSA, AP meaning alkaline phosphatase, and then QMSA meaning quinone methide signal amplification. And it uses QMs, which are quinone methides that are dephosphorylated by the AP, producing a reporter that is able to quantify again where tissues are cancerous. So the methods of this study, they basically took different types of QMs and they're able to be dephosphorylated by the secondary antibody that has the alkaline phosphatase bound to it. So just like Western blots, you have a target gene and then your primary binds to that and then your secondary binds to your primary. And this allows us to be able to have alkaline phosphatase in this reaction. And then once the phosphate is cleaved off, you are left with a molecule that has to have a leaving group. So the way that they looked at this study was two different types of molecules that had two different leaving groups, one that had two fluorines coming off and one that had one fluorine coming off. And once the leaving group comes off, they made a really reactive molecule and it's able to react with nucleophiles, whether it be on the primary antibody or if it reacts with media and then it's destroyed, or what we want, it binds to tissues, specifically cancer tissue, and is able to report or quantify, allowing us to measure where and how much cancer is present. So the way that they did this in their results, they took the AP-based card immunohistology of the two different types of QMs, so the difluoro meaning two fluorines and monofluoro meaning one fluorine, and they were trying to see like which type of QM produced better results. But they also were looking at this under different pHs. So the first box here shows the control, and they just wanted to make sure that it didn't stain where it wasn't supposed to. And then they went up in pH, so pH 7, 8, 9, 10, and 11. And you're able to see that like there's a little bit of staining, but not too much. And then in pH 8 and 9, there's a lot more staining because that is where AP is the most reactive and that's its ideal pH. And then when you compare this to the monofluoro, you can see that there's a lot of on-site binding, which is what you want, but there's also a lot of off-site binding, which is not what you want because you want to be able to specifically see only where the cancer is. So they found that difluoro is much better for this, and it also just has more clear binding at pH 8 and 9. So some practical applications.
So these are some primers that we created using the DNA sequence for alkaline phosphatase that can be used in PCR or polymerase chain reaction. So the forward primer is shown at the top here. It can be used to initiate the synthesis of a new DNA strand during PCR. And then the reverse primer can be used to start the synthesis of the complementary DNA strand in PCR.
And then some clinical takeaways from all of this. By understanding more about the function and structure, we're able to understand abnormalities in AP and in the isoenzymes. And one common mutation or abnormality is in a disease called hypophosphatasia or HPP. And this is caused by low serum alkaline phosphatase levels, resulting in abnormalities in tissue nonspecific AP. And this can lead to difficulties in bone mineralization, also with lots of other detrimental effects. And they have linked this to 260 different types of mutations causing HPP.
These are our references. Thank you.